Come in, everyone. We are waiting for you. The program is about to start. Thank you. Come in, ma. We are waiting for you. Come in. Thank you for coming. The turnout in the auditorium is very low. Please come in. Once again, good morning, ladies and gentlemen. Good morning, everyone. I believe this weather is not affecting us. No, it can't affect us now. We are too much for that. Please, can we please stand up to shake out the cold and welcome like 2, 3, 4 people next to you. Say, good morning, my neighbor. Good morning, my neighbor. Good morning, my friend. Just let us shake out the cold. Let us shake out the cold with a smiling face. Yeah. Good morning, everyone, once again. Please let's have our seat. Let's have our seat. Today, 18th July 2024, is another seminar day, institutional seminar day. Let's put our hands together. Let's put our hands together. It's too early for this code to affect us. Let's put our hands together. And I welcome everyone to this great seminar and impactful seminar that we're going to be having today. And I believe it's going to be very, very impactful as we pay much attention to every word and to every presentation. So this great institute is going to be an impactful one, and to everyone it's going to be an impactful one. Welcome once again. Now we are going to be having the national anthem. After the national anthem, the pledge. Please, let's rise on our feet for the national anthem. Now the third, please.
Bless Nigeria, my country, to be faithful, loyal, honest, stand and care of all my strength, to defend thy duty and uphold our so
Let's thank the general once again in honor of our nation. Yeah, we have important dignitaries in our midst. And before we start, we are going to be introducing them one after the other. And they are the IMC members in our midst. The first citizen of this great institute and person of Dr. M.L. Atonda. Let's put our hands together for him. He's on official duty. He's not here with us. He's on official duty. Still on my list, we have Dr. O.A. Adetula. Dr. O.A. Adetula is Director of Research, Chairman, Research Review and Planning Committee. You are welcome, ma'am. Also in our midst, we have Dr. V.P. Ume. Dr. V.P. Ume is the Director of Research and the Chairman of the Parliament. You are welcome. And also in our midst, we have Dr. H.A. Akintoye. Dr. Akintoye is the Director of Research, Director of Education and Law Research. We can do more than that. Please take the hands together. Dr. Akintoye, you are welcome, sir. Still, we have in our midst Dr. Ayo Olajide Taiwo. Dr. Ayo Olajide Taiwo is the Director of Research. Sorry? L.O. Yes. Pardon me for that. L.O. Olajide Taiwo, Director of Research and Public Relations Unit. Let's put our hands together for him. Then we still have in our midst, we have Dr. Mrs. Adeoye. Dr. Adeoye is Director of Research, Health, Family System and Extension. You are welcome, ma'am. Thank you for coming. I'm so sorry, ma'am. Still in our midst, we have Dr. Mrs. Esu Abalola. Dr. Mrs. Esu Abalola, Director of Research, HOD Citrus Products and Development. Ma'am, you are welcome, ma'am. You are welcome, ma'am. Oh. Still while introducing Dr. Babalola, Dr. Mrs. D.O. Babola Woting, and she is the Director of Research, Health Information and Documentation. That's perfect. You are welcome, ma'am. You are welcome, ma'am. Okay. Now the IFC has been introduced. And let me give this information to us. For the page of the NIOT, if you can bring our Bible out to jot it down. We have the pages for NIOT that we can have all what we are doing on the social media. We have the Facebook. For the Facebook, we have at NIOT NG. At NIOT NG. Please put that information down. And again, for the Telegram, we have at NIOT Research. And for the Twitter, we have at NIOT NG. Now, for the live stream of this program, we have it on YouTube at NIOT Research. Do we get that clear? Okay. Thank you so much. To the business of the day, to introduce to us with a clap ovation, and let's please rise up to honor him the chairman of today, for this great seminar. Please, let's please rise up to honor him. And with the clap ovation, I will introduce to us the chairman for this program that we are set to go in for. We have Dr. B.C. Ume and Chairman. Chairman, sir, you are welcome, sir. Dr. Mrs. Karim Katie, you are welcome to our meeting. She's one of our guests. You are welcome. We appreciate your coming. Thank you so much, ma'am. Now, on the program, I would like to introduce to us the presenter for the seminar for today, in person of Dr. Keita Egberumbe. So sorry about that. Thank you so much, ma'am. The reading of the citation for Dr. Egberumbe will be presented by Dr. Diron Olaleye. That's what I did for the reference. If you can come and have the floor.
Good morning, everyone. Standing on the already established protocol, I would like to urge Dr. Kenny Egberumbe to please be on our feet and step forward here while I read my presentation. You're welcome. Dr. Rafiat Kenny Egberumbe was born on the 7th of January in the 70s to the family of late Alhaji L.A. and Alhaja R.A. Egberumbe from Abeokuta in Ogun State. She started her academic pursuit at Sacred Heart Private School, Onireke, Ibadan. From there, she proceeded to St. Louis Girls Grammar School, Mokola, also in Ibadan, for her secondary education. Dr. Egberumbe had a BSc in crop protection and environmental biology from the prestigious University of Ibadan in the year 2000. She proceeded for her MSc in environmental biology in the same university, and she bagged her MSc in the year 2006. And in the same university, she bagged her PhD plant ecology in 2016. She did a National Youth Service Program at the International Institute of Tropical Agriculture, Ibadan, in the year 2001. Dr. Egberumbe started her career at the National Food Reserve Agency under the then Federal Ministry of Agriculture and Water Resources, Abuja, where she rose to the position of Senior Planning Officer, Agriculture, before she left the service of the National Food Reserve Agency to join NIOT in 2009. She rose to the post of a Senior Planning Officer, and she joined NIOT in 2009 as a Research Officer 1. And since then, she has risen through the ranks to her current position of Assistant Director, Research. Membership of professional bodies. She is a member of the Organization for Women in Science for the Developing World. She is also a member of the Ecological Society of Nigeria. She is a member of the Wheat Science Society of Nigeria. She is a member of the Horticultural Society of Nigeria, a member of the American Society of Plant Biologists, member Nigerian Conservation Foundation, and a member Nigerian Society for Plants Protection. Awards and merits. Dr. Egberumbe, during one of the Asuri end of the year party, precisely in the year 2019, was awarded the most research scientist. And that's very, very, very good. And she also during a degree program in the 1998-99 session in the University of Ibadan. She was given a certificate of merit from the Dean of our Faculty of Agriculture and Forestry as the best student in cowpea production for the year. Some training workshops that she has attended. She attended the management training on personnel relationship conflict management held here in NIOS in the year 2022. She attended a one-day training workshop on intellectual property management, patent information, search and patent drafting procedures for researchers and inventors held at Colossus Hotel Lagos in 2022. She also attended craftsmanship workshop organized by Genetic Society of Nigeria held at Adipo Ali Hall, More Plantation in Ibadan in the year 2022. She also attended a plagiarism detection application for MFP, MPhil, MPPhD, and PhD students while she was on a PhD program at the University of Ibadan in the year 2015. She attended the University of Texas, AM, USA, in collaboration with the University of Ibadan, Faculty of Agriculture and Forestry, Department of CPEB, a workshop on breeding for resistance to plant disease causes in the year 2011. She was also at University of Port Harcourt, Nigeria, to attend a workshop on electronic library resource training for research for life and C programs conducted in the year 2010 in collaboration with Institute of Development Studies UK, Cornell University, and Itoka, South Africa. As part of our ad hoc services here in NIOT, she was a member communicated drafting committee in the year 2023. She was an internal assessor on senior staff promotion in the year 2023. She is a member and secretary of Project One. She is a focal person for citrus program for NIOT newsletter. She is also a member committee on compilation of NIOT technologies in the year 2019. She was also a member of the Hot Consult Committee in 2019. She was a member and secretary ad hoc committee on patenting of NIOT technologies in 2019. He is a coordinator of interns and youth club members in Citrus program 2018 till date. Services rendered outside the institute. He is a reviewer in Nigerian Journal of Ecology 2021 till date. He is a reviewer with Agricultural Science Research Journal 2022 till date. He also served as a member of the local organizing committee, Ecological Society of Nigeria in 2022. Also, as part of our community service, she was a resource person at the Institute Radio Program, Ishiola, in March 2024, where she spoke on the topic, soil fertility management in horticultural crop production, the role of nitrogen fixing plants. She was also a resource person during the training held on mango value chain organized by NIOT in 2019 at the AGP Ogumosho Zonal Office, Ogumosho in Oyo State. Other community service, she is a member and donor of horticultural materials to Akebile Landlords and Landladies Association in Akiyele local government. He is a member and she participates in environmental sanitation activities in Ring Road Landlord Association since 2010 to date. He used to be an examiner with West African Examination Council 2002 to 2006. Dr. Ikperupe was also a member of the Federal Road Safety Corps during her service year in 2001. Dr. Rafiat Kehinde has over 40 publications appearing. Thank you. Appearing in reputable international and local journals and conference proceedings. I hereby present to you Dr. Rafiat Kehinde.
Let's put our hands together more. Dr. Duran Olaleye, thank you so much for that great recitation. Thank you so much. Let's appreciate it. Let's appreciate it. The next agenda is to introduce to us the rapporteur for today and person of Mr. John Ikoro. Mr. John Ikoro, thank you so much. Welcome to our meeting. At this junction, I will hand over the mic to the chairman of today to continue with the program. In person of Dr. V.C. Omek. Thank you. I would like to welcome to our meeting one of our guests from UI, Professor Awodoyin. You are welcome to our meeting. Thank you so much. The chairman, sir.
Thank you, see. Great job. And before I continue, let me welcome again who are not mistaken, the supervisor. Public for communication. That's I can say the audit is here. One where discuss. It is not around.
No, last time.
Our presenter, as you have to, is a quiet lady. You want to go, she's around. Sometimes when I see her, I say, are you in this room? You say yes, and she's always here. So I present to you Dr. Werombe to give us a talk on Sesbania, pachycarpa, a Ruderal and agricultural resource. I believe I join this. Get me.
Good morning, the ED and CEO of My Health in Essential, all management members present, invited guests, ladies and gentlemen. The title of my presentation is Sesbania pachycarpa, DC, that DC is the CANDO, that's the authority, a rural and agricultural resource. Sorry for that short break. I'll be making my presentation following that outline. The weed concepts. We have different ecologies, and these ecologies differ with their vegetation. For example, the forest areas are predominantly trees and few woody climbers. The savannas are mostly dominated with grasses, few trees, and some shrubs. The mountainsides and the seashores, they are the sites for the habitats for the plants we call weedy species because of the openness. And this openness is as a result of disturbance, either natural or man-made. Natural, for example, we have landslides, volcanic eruptions, and earthquakes. Therefore, deliberate actions, we have man clearing the land for production of food, clearing the land for road, and mining minerals in the innermost part of the earth. Since vacuum cannot be created in nature, these plants move in to occupy the space, and they become the pioneer of a process called secondary succession. That's the revegetation after a disturbance. The process of the vegetation of land denuded or stripped by agricultural activities is what we call weed infestation. A weed in a general sense is usually described as a nuisance, and unwanted plants in human control settings like farm fields, lawns, and all those areas. More specifically, they are used to describe plants or native or exotic that reproduce aggressively. The term is no longer controversial in that each scientists define what it means as a weed in different situations. And so the word meets the practical use of the user of the word. Considering the possible benefits of weeds, they have been described as plants whose virtues have not been discovered. There are about 2.5 million flowering plants in the world, and all these are grouped inside 450 plant families. Of these, 250 are so much interfered with man, and they have been grouped specially as weeds. Two families are almost form half of all this population. They are Poaceae and Asteraceae family. Poaceae house many grasses like Panicum, Maxicum, Paspalum, and all the likes. While Asteraceae is a family to Titonia diversifolia, Promolena odorata, Ageratum, many broadweeds. Of all the pests of agriculture, from vertebrates to invertebrates, the effects of weeds are the most underestimated. We don't usually take them serious because they are always there with us. We see them every day. But they do their damage silently, without drama or show off. You won't compare weed infestation with having a problem of yellowing disease or wilting. You quickly jump to action to curtail it. But weeds, you say they are just there. If you have the time, you can weed, but they are there silently doing their damage. Most of these weed definitions have been faulted based on the fact that not all weeds are poisonous and useless as we think. And until we find uses to them, we don't call them weeds. Mulligan identified two groups of weeds based on their location. The agrestals are the farmers' real enemies. They are the ones competing with our crops on the field for land, for water, resources, and all that. While the ruderals are the public nuisances. They are the ones on the roadsides, around the lawns, recreation centers, and so on. In Southwest Nigeria, legume shrubs like Tephrosia and Senna, they are fast disappearing from our natural ecosystem. And the uses of these plants, the advantages they bring are also lost with them. Recent invasion of some roadsides with Sesbania may be a grace in restoring natural fallow management. Sesbania is an annual tropical plant with origin traced to Africa and is especially endemic to the Sudan and Sahel ecologies. That's the taxonomic classification. It belongs to the family Fabiaceae. This is a picture of Sesbania plants on the field. The introduction of Sesbania to this ecosystem might have been through importation and distribution of food grains and livestock from the Republic of Niger. In Niger Republic, the seeds are consumed, boiled with other condiments. The flowers are eaten in salads. The fresh pods are eaten as vegetables. The leaves are used in treating malaria and mouth infections. Extracts from the roots are known to treat tumors, and the mature stems are used as firewood or for light construction. So it's a complete package to man. This is a picture showing Sesbania with other plants. Sorry. Yes. This is another picture of Sesbania. This is Sesbania. They go as high as 4 meters high. With other plants, it was taken along Ipapo. This is another plate showing Sesbania. This is Victoria by the side. It was taken around Oshogo Expressway. While working on this plant, I did a survey throughout Southwest Nigeria to check the distribution and spread. And on a few occasions, I had opportunities of meeting with local farmers on site. And I discovered they have been using this plant on their own. They are suspected it has something to do with soil improvement, and they cut from roadsides and use on their farm on small scales. So it wasn't entirely new to some of the farmers. The beneficial nitrogen fixing potentials of some plants can be harnessed in the farming system to restore nitrogen to the soil and reduce fallow period, thus increasing crop yield and solving the problem of food insecurity. Also tapping into the atmospheric nitrogen will reduce external inputs in form of inorganic fertilizer and saving the country huge investment on fertilizers and protecting the environment against pollution. It has been reported that half of what we use in our field as inorganic manure is what the plant actually uses. The remaining 50% is lost to the environment. Bola reported that this plant fix the corresponding nitrogen content within 45 and 60 days of growth. This presents legumes as an alternative to the application of inorganic fertilizers. This is to compare Sesbania with Sphilanthus amarus, that's Enyolobe. I have both plants that you can check up later. They look quite similar as seedling stage, but they are very different. I also have the seeds on the table. Those are seeds of the plants. Sesbania have exceptionally vigorous symbiotic association with the Rhizobium bacteria, and they are presented with nodules. They also have extensive taproot system that serves as nutrient pumps, recycling nutrients that have been leached into the soil. This plate is showing a slide of Sesbania with the nodules. These are the nodules on the lateral root. Those small, tiny circular things are the nodules responsible for nitrogen fixation. And the appearance of nodules on plant does not necessarily mean the plant is fixing nitrogen. You have to observe. Make a cross-section of the nodules, detach it from the roots, make a cross-section using a sharp instrument, and observe the color. Colors like white, green, or gray shows the nodules are not effective. They are not fixing nitrogen. But when you cut and you observe the color, you see any coloring from pink through red, it shows the nodules are active and they are fixing nitrogen. And that pigment, that red coloration, what is responsible for it is leg hemoglobin. Like we have hemoglobin in blood, we have that for plants. And what they do is actually a protein. They carry iron molecule and they help transport oxygen throughout the root system. For possibility of integrating Sesbania into conservation agriculture, there is need to understand some of the aspects of its ecology, like seed germination, early seedling growth, and the green manure potential. And so, when you plant the seeds of Sesbania, they don't readily germinate, which means they have seed coat dormancy. And I used different methods to break this dormancy. The one that gave result was with conch H2SO4. And I varied the duration at which I exposed the seeds to the acid and planted in Petri dishes. Sowed the seeds in Petri dishes. These are the varying durations of the exposure to the acid, up to 2 hours. And the result shows that for maximum germination, you can actually treat this seed. You can treat the seeds for up to 50 minutes, although the seeds tolerated acid treatment up to 2 hours in conch H2SO4 without any damage to the resulting seedling. How then do we encourage farmers to use this machine, knowing fully well they may not have access to the acid? So I thought maybe we could break the dormancy and give to farmers. So I did a study to find out how long the viability will stay. So over time, I was testing the seeds out to check the viability. And the results showed that you can actually, once you break the dormancy, you have to plant within 1 month. Otherwise, the germination drops along the time. You have, we had 73 here at 4 weeks. So after 4 weeks, you don't have to keep waiting. You can just plant within 1 month. Then I also kept some set of trees for up to 4 years, trying out the viability at a particular time each year to find out how long the fresh seeds will stay viable. And I discovered they can actually stay up to 4 years. These are the freshly collected seeds I was trying out every year. I did a study to find out how Sesbania will respond in various levels of soil fertility. And I deliberately used NPK to vary this fertility from 0, which is a control, up to 75, representing high. I used the marginal soil, which means it is deficient in most nutrients, as a starting point, and it was a pot experiment. This plate shows how Sesbania responded to fertilizer application 6 weeks after the application. This is the control trying to survive as a plant, not as a nitrogen-fixing plant now. This is the high treatment, this is medium, and this is low. I also did a study to check how Sesbania will nodulate in these varying soil fertilities. What I discovered was that with nitrogen-fixing crops, if they grow in very marginal soils, in very poor soils, they rarely nodulate, because at that point, the plant is trying to survive on its own as a plant and will necessarily not fix nitrogen in such conditions. Even when the fertility is too high, they shut down their normal response to bringing out nodules, because the plant gets a signal that, look, this soil is fertile, don't stress yourself. And you don't normally get high nodulation in high fertility soils. So this result, we had as you increase the fertility, you have reduced number of nodules produced. This was the highest, and it was from low fertility treatment. I also did a study to find out the green manure potentials these plants could offer, and I used Amaranthus guensis as a test crop. The variety was NHAE. This was a pot experiment in CRD and 3 replicates, and I varied the amount of days of planting Sesbania. So I have a range from 0, which is control. Those are pots devoid of Sesbania plants, up to 75 days. And this was the result. If you grow Sesbania plants for 45 days, because with nitrogen fixing crop, what they fix remains within the plant, not that they dump this nitrogen into the soil. We have just 2% leaking into the soil. So all the benefits of the nitrogen fixation remains within the plant, which means you have to turn back. You have to cut it to pieces and turn the material back to the soil, allow it to degrade before you get the benefits. And so at 45 days, we had the highest growth parameters for amaranthus at 6 weeks. 45 days gave the highest, and it was significantly different from control. When I harvested this first set, I planted Sesbania again. I planted amaranthus again to check if there will be any residual effects that the plants can enjoy from the manure. And so the same trend was noticed. At 45 days, we had the highest growth parameters, and it was significantly different from control. I also did a soil test to find out how nitrogen is within these ranges. We started from 0.01, and nitrogen increased across the treatment. This is a summary. Sesbania has inherent seed coat dormancy that can be overcome by acid scarification for at least 50 minutes, although the seeds tolerated acid treatment up to 2 hours without damage to the resulting seedlings. The tolerance of acid treatment for as long as 2 hours may indicate that this plant will tolerate acid soils and therefore will be an ideal pioneer plant to restore degraded soils. Acid scarified Sesbania seeds can be stored for 4 weeks before the viability declines and become non-viable. However, untreated seeds that were stored in room temperature, in envelopes in room temperature, for 4 years were still as viable as the freshly collected seeds. Sesbania responded positively to nitrogen fertilizers. Growth and nodulation of Sesbania are retarded in soils with very low fertility. Therefore, they may need a starter, nitrogen fertilizer as a starter on nutrient-depleted soils. Also, Sesbania plowed in and screened manure at 45 days after sowing benefited the test crop, which is amaranthus here, most in terms of growth performance. The second crop did not benefit much from this green manure, probably because Sesbania is rich in nitrogen than lignin and polyphenols, and they may not contribute to long-term enhancement of soil nutrients and organic matter. But it can be assumed that this amount is limited. Continuous use of this material will ensure modest amounts of nitrogen for the soil, thus increasing the yield of subsequent plants and reducing the effects of pollution. The reduction in inorganic nitrogen fertilizer inputs by adopting this green manuring will reduce environmental degradation in 3 ways. You know, I said earlier that half of what we use as inorganic manure is what the plants actually use. The rest is left to nature. In 3 ways, nitrogen can be lost through surface water runoff. And we get these materials deposited in water bodies nearby. Because of this rich source of nutrients, algae and other low-growing plants make use of these materials and they grow. They increase in population. In the process, they use up all the oxygen in the water and other life is gone. So we have only these plants surviving, and such water bodies are called dead zones or algae bloom. That process is eutrophication. If you don't have it lost in that way, when there's rainfall, they will gradually move through the soil profile and get to the underground water, pollute it before ever using. That's another way. And the third and the most important one is the emission of a gas called nitrous oxide. That's N2O. Nitrous oxide is a very potent greenhouse gas. When you inhale it in small doses, there's the feeling of calm and relaxation that you get. And so it's called laughing gas or happy gas. When you increase the dose at which you take in nitrous oxide, you start to get dizzy. Later, you have some hallucinations, there is mental disorder and death. When you take it in small doses over a long time, it causes infertility in both sexes. For females, it stops ovulation. There's a part of the brain that it affects that it stops that mechanism and the female stops ovulating. For men, it affects the motility of the active sex cells. Nitrous oxide is the effect, the damage done can be treated when you treat early, within the first few weeks of damage. If you miss it that period, then they become irreversible and permanent. And the treatment is with cobalamin, that's vitamin B12. It's administered intramuscularly up to a period of 12 months, and that will reverse the effect. But if you miss it early to treat, it becomes permanent and irreversible. That's the harm to man. You can boost your system, fortify your system, if you're likely going to be exposed to laughing gas by consuming B12-rich foods. You get this vitamin from liver or either cow or sheep. That's the highest source of B12. You can also get cobalamin in beef, dairy products, seafoods, and the likes. From fruits, you get B12 in oranges, bananas, and apples. From vegetables, you get in spinach, potatoes, and also in mushrooms. They are good sources of B12 that you can use to boost your system. That is the damage done to man. To the environment is a greenhouse gas. And like most greenhouse gases, what they do. Greenhouse gas, they got the name from the structure we call greenhouse structures. They look like a house and they are housing green plants. So that's why they call them greenhouse. They look like houses and green plants grow inside. So they call them greenhouse. The same thing, what these structures do is that they trap heat. And that's exactly what greenhouse gases are doing. They act as a barrier, as a blanket, preventing the exchange of heat and other gases. And so the heat we generate is no longer escaping into nature. That is why we are having a problem of global warming and climate change. Greenhouse gases, I'll talk about 4 that are from natural sources. There are about 10 of these gases that pose this problem. Nitrous oxide is one, N2O. It contributes 6% to the total global greenhouse gases, but it has a global warming potential of 298. That's the ability of the greenhouse gas to cause global warming. That figure for nitrous oxide is 298. Carbon dioxide is another greenhouse gas. That's the gas we exhale. We take in oxygen and we give out CO2. CO2 is another greenhouse gas and it has the highest contribution to greenhouse gases. CO2 has 76%, but it has a global warming potential of just 1. Methane is another greenhouse gas, CH4. We get that from animal production. In the rumen of these animals are special bacteria called methanogens. They are the ones that act on the ingested food to degrade and digest it. And in the process, the animal belches. That gas releases methane. It's colorless, it's odorless, it's highly flammable, relatively safe to man. But when you start ingesting in little doses over a long period of time, then you come down with memory losses and vision problems. That is the problem of methane. For CO2, we can actually mitigate it by planting more trees, afforestation, because trees, like plants, they have the ability to mop up our carbon emissions and give us oxygen in return, especially bamboo, Bambusa vulgaris, Opam will absorb 5 times what normal tree will absorb and give you 35% more oxygen. So they are very important trees. If you are lucky, you have a group of Opam in your neighborhood, pamper them. They are doing a lot to help us mop up all these emissions and give us better air, more oxygen. The last greenhouse gas I would like to talk about is water vapor. That's the steam we generate from cooking or boiling water. It's a greenhouse gas and its composition is variable in the environment. It has a global warming potential of 0.005. We transitioned from global warming to global boiling July 2023. And that was the hottest month ever recorded in the history of our planet. Global boiling is the transition from global warming and is a period characterized with intense heat waves, extreme weather conditions, and climatic disasters. We are in this period and it's expected to last for a period of 5 years, according to UN. So by 2028, we'll be in for another weather surprise. What we can do is we have to mitigate all these effects on our own part. We have to start exploring, start developing and adopting eco-friendly methods in solving our problems. Whether they are issues of soil, pest, or disease, there are friendly methods you can use that will be safer for you and your environment. Greenhouse gases are not the only pollutants in air. We have a lot from microplastics, we have from burning fossil fuels, we have various effluents from various industries, and all these pollutants are released into just one planet. Remember, this is the only planet for now that supports life and our growing population. Why won't we guard it jealously? Why won't we protect it? Why won't we conserve what is left of the ecosystem for us and for generations yet to come? I'd like to thank Almighty God. I call him the gracious, the merciful. God has been merciful. To our ED and CEO, we thank him for coordinating and directing our affairs of NIHOP. The management member, we thank you for your support. Head of Program, Citrus and PDP, thank you for your encouragement. Head of Program, Citrus, thank you for managing us in Citrus programs. HOP, Project Development, thank you for your contributions. Staff of Citrus Program, thank you for your cooperation. My mentors in UI and NIHOT, thank you. Professor Agbodoyi has been my mentor since 1999. I always go and come back to him. Thank you for long years of training. I'm grateful. To my family and friends, thank you. My wonderful audience, thank you for your attention.